Okay, hopefully this will be useful, guys. But so I'm going to talk about biobank construction for translational research and just some conflicts of interest. And disclaimer, I don't know all the answers, but I've been able to create 2 biobanks at Yale and UCLA from scratch due to kind of limited or prior limited like institutional efforts. And it's led to really, 200 patients at UCLA, and at Yale I was responsible for putting it together. We collected about 1,500 patients of all GU tumors. It's still in existence, so maybe David, you can harness some of this, but we built everything from scratch. And most of the collections have been surgical, but that's basically because I haven't had really willing med-onc partners who really kind of cared about translational research. So if you know anyone, please send them my way because we really need someone here. And I made a lot of mistakes, so hopefully you can learn from some of my mistakes. I'm happy to share them with you. But clearly, when I started, we just had like this nice picture from an old talk, bench and clinical research, just showing kind of how where translational research kind of fell into place. But the figures have changed, and now there's these really large, expansive, obviously T1, T2, T3. I always have to go refer to it, but I think all the research on bench to basic science will fall in this T tier to T1 to T2 range. But really, you can't really do patient-centered research without patient samples. Definitions have changed, but really translating care to the clinic really relies on the bench and access to specimens. So I think specimens are the cornerstone of this type of research, especially with biomarker work, and really obtaining really high quality, well annotated specimens through an organized, comprehensive approach, I think are really critical. So I think when you guys are starting out, I didn't know who was going to be on the call. So a lot of you guys have started to establish yourselves, but I think you always have to say, do you really need to go through the hassle of really chasing down specimens through your institution or practice? There are plenty of other ways to get access to specimens without some of the hurdles, such as, you can collect de-identified specimens, which have less red tape. But once you start getting into patient demographics, PHI becomes a challenge. So, you can get commercial resources. If you just Google, you can find plenty of specimens available from companies that are willing to sell fresh or frozen specimens. And most of them, if you look at some of these companies, a lot of them have shown they've done it. They partner with institutions and they have like either tissue microarrays, they have access to, fresh tumors, and they're available for, purchase. But there are a lot of NIH research resources, various banks sponsored by the NIH or NCI, depends on the disease type, and they range from specimens collected and shared with the NIH, or even PDX. There's a PDX library. Instead of making your own, you can basically just purchase someone else's. And you can look, here's another one, the specimen resource located. You can look at kidney, you can find, different types of things, kidney leiomyosarcoma, things that might be useful. You're not going to have, hundreds of specimens, but they have enough available that could be potentially useful. And then obviously NCTN, this is the Clinical Trials Network Navigator. So I put this up here just to show that, there are trial specimens that are linked to a lot of very good clinical and data. The issue here is a lot of these, the network like SWOG, you have first crack at it is built in some of the correlative plans. And after about 6 months after the trial results, it kind of gets released for broad use. I'd say this is actually really a great resource, but it's really hard. So I knew Brian was going to be on. Brian's CALGB BEV interferon trial was really an amazing, effort. And Hyung Kim wrote a grant based on it. And by the time the grant was funded and he got access to the specimens, BEV was not even really used anymore, and CALGB wasn't even a cooperative group anymore. And Brian still looked the same age. So, things change, other things don't. But Hyung will tell you for 5 years he was paying a statistician and then finally got access to the specimens and they wanted more money. He's like, what have you, for 5 years I've been paying you and you haven't done anything. So it's a little bit of a challenge. And Ritesh, if you speak to like Jonathan there, he had I think he had like an Avastin bladder cohort from, I think it took him like 4 or 5 years. He can tell you about his journey there. It's just really challenging to work with a cooperative group cohorts. Here is the NCI patient-derived models repository. Microarrays, there may not be that much stuff available, but there could be something useful for your research. And then why build it? Can you share? So of course, sharing specimens across institution, if it's not commercially available or federally, can you basically, maybe you don't need to build. Can you put together a cohort of maybe pancreatic metastasis with Brian or Jim Brugarolis, and you look at that instead of actually going through the rigmarole of building something yourself. Obviously, you need to establish like a MTA or CRADA. There are clearly some regulatory hurdles. We're trying to do this with Payal Kapoor, look at small renal masses and anything with identified information like imaging that we're trying to share with Ivan Pedrosa. It's like 8 layers of red tape. So there are hurdles here, but you can get it to work. And then obviously you don't need to reinvent the wheel. Where you're going, if there are institutional resources in place to help you with your mission, could be very useful. There are existing databases or biobanks potentially. So when I came here, there was an nephrectomy database of 4,000 patients. Clearly, I can build my own, or I could just follow what they have for this clinical database. But there wasn't a database of resected specimens, so I had to build that. Are there protocols in place to help you implement your approach? Well, we have a procurement, at Yale, there was a procurement team run by a guy named David Rim, but he would not put together an institutional biobank because in his experience, it was never used and no one wanted to use things collected different ways than they wanted for their own specimens. So we were able to harness that resource, but clearly there was no institutional centralized banking. Same thing here. There was kind of a disorganized, semi-centralized bank, but there was no really rhyme or reason what they would collect and how they would collect. So, first things first is, just you can retrospectively see what's available. The good thing is there's everyone who's participating, any academic center, they're like accredited by the College of American Pathologists. And like paraffin blocks, they have to be kept about 10 years. IHC slides. So, you have a PowerPath system, EMR search. You could put together like, institutional series at Sloan Kettering, 15 years of brain mets. You'll probably find like 50 resected brain tumors. The serum and plasma clearly are not stored or banked or frozen. What I have heard around from a lot of people, including here, is these take up a lot of space. So they're starting to throw away older specimens, which is really a little bit disappointing. So we're actually going through and keeping ones that are over 15 years old, and we have like a whole, centralized area now we're going through. Prospective infrastructure. Well, do you have a universal consent? And can maybe you be added to that universal consent? Why start your own protocol, whether you could just be part of an institutional effort as a collaborator? Again, the team, you can be linked to that procurement team. Maybe they charge you. One thing is their phlebotomy lab team, or maybe if a patient's going for an infusion, couldn't they actually draw blood for you at a standard of care stick? It was great at Yale. We actually had the lab willing to, I gave the patients the tubes, they'd show up for their blood, and they would have the blood drawn at the same time. Here, I don't have the luxury here, so we collect it at the same time as surgery. I have usually a moody anesthesiologist, but we usually get through it. And can you actually use the specimen processing services? Like if there's a clinical trial team and they're processing blood, can you actually just give them the specimens rather than hiring someone yourself? Again, don't reinvent the wheel. All institutions obviously have a lot of red tape, IRB protocols that are needed to be followed. However, every institution is different. There might be a cancer committee. We have to go through that here. An institutional biosafety committee, you have to go through that here too. You have a protocol committee. I'm just gonna put this on a different screen, guys, because my fellow wants to listen in. So I'm gonna have if you don't mind, just gonna change it over to the other screen. Okay. Okay. We also have a biosafety committee. And for biological specimens, they want to know how you're going to handle that. Is it going to be used safely? Protocol review committees also, IACUC committees for PDX work. There's a lot of red tape, but once you have it in place, it's great. A lot of places have a tissue advisory board. Fortunately, kidney, there may not be as many people interested in that at a lot of institutions. So sometimes it could be flying under the radar. But I assume at like a place like Sloan, when you have like 15 different teams, maybe everyone wants a piece of, a specimen. Now, waiver of consent. So the important question is, do you really need clinical information? Can you just have like information about basic stuff like size and type? And if that's the case, you're in good shape because you have less red tape. If not, you might need to do prospective specimen, consenting for every patient. And that becomes a little bit tough because, when's the best time? A lot of times you see people before surgery, they're nervous and you have some research coordinator coming on, not necessarily ideal. So, basic demographics for this specimens that get a waiver of consent, I think are really useful. Obviously, informed consent's needed for prospective. I would definitely, on a consent, try to make it as universal as possible, have as much flexibility as possible. Thanks, Brian. And then also try to allow for different time points and also different clinical conditions. So my protocol at Yale was specimen collection of GU cancer. It was like hereditary syndromes and GU cancer. So I was able to collect a lot of patients that, maybe weren't like just necessarily cancer. There could be other things like polycystic kidney disease. We have other patients who might have like, SDH, but didn't have a tumor. So I was able to get like a nice broad consent. And then obviously flexibility to share specimens with outside commercial institutions. It was really useful to have this available as we have a lot of contracts with different companies. And you need paper consent. So yeah.
Right, is it okay to ask questions sharing?
Sure, yeah.
Can I ask for the sharing with outside commercial institutions, is that typically on a per outside entity sort of basis, or is that something we can have like broadly in the consent?
I have it broadly. It just says your specimens will be used broadly with commercial entities that may be for profit. And that's how the Yale one is set up, is the way I've set it up here. So really there's no, if it's a biobank, it's collected, as long as patients may know their tissue might be like sold for cancer research to benefit society, you don't have to re-amend the protocol.
Okay.
Yeah.
That's helpful. Thanks.
But doesn't that, when you make the agreement, then you'd have to specify, like we've kind of gone through this a couple of times where we have to then specify exactly what is the use going to be and then the ownership going to be, right? From the back end, or no?
Well, you might have some contracts with that company, but if you collect the specimen broadly for your bank, the patient donates to the bank and you don't have to go and reconsent them.
Okay, got it. I got it. Yeah.
So you're basically sharing existing specimens, or, if you have a patient prospectively, say, hey, I'm going to start collecting urine, it's collected for the bank, and by the way, I'm going to give some, to another company as well.
Sure. Yeah.
Do you need paper consent? I use DocuSign here. It's been very successful. A lot of institutions won't use it, but during the pandemic, it's been great, and we've adapted it here. So a lot of times I have a video. I send patients the video in my chart, and then they respond they're interested in it, and I send them a DocuSign. So they had someone talk to them about the protocol, and then it's not like a therapeutic protocol. You can't put someone on the latest and greatest like HDAC inhibitor by DocuSign, but I can collect their poop, by DocuSign. So patient recruitment, obviously you might need some flashy thing on my, if you send them or information or a handout in clinic, we have a nice kind of handout over here. It was my career goal to have a recruitment flyer with pieces of poop. The poop emoji, this is something you might never see again, but we have a stool microbiome. I collect, a part of our tissue repository, our biospecimen repository. We can, have this on there. And I did something like this at Yale. Here we have our own special consent for this protocol, but again, make it as broad as possible. And a nice emoji goes a long way. There's some of my templates for different, a small biopsy trial, a sestamibi trial, all those different things. Institutional guidelines, obviously you need to know how much you can take. Most places like 3 to 6 teaspoons, and some places like 50 cc's. I think it's very tough. Every place has a different definition of remnant tissue. So like, obviously, if you're taking out a large giant piece of tumor, you're not going to use all of it, but a biopsy is definitely a different story. So additional cores, it's a big issue. How many cores does someone need? Like, I'm going to take 5 cores, and if I take 2 excess one, it's not really remnant tissue. Okay, so you have to, for like some protocols, calling it an additional core, it makes it a higher level of safety for some institutions. Okay, because you can't just keep on biopsying indefinitely. And the other issue is that if a specimen's removed, for standard diagnostic evaluation, surgeons can't just cut it and take it. There was a case at Yale, which David Rim could tell you, that a surgeon cut into the specimen for colon cancer, and they did the pathological diagnosis, and there was no tumor there. The question was, did the patient really have colorectal cancer, or did the surgeon, were they just greedy? But things like fat, like if you're going to do a project on looking at like perinephric fat, that's discarded. That's discarded tissue. We have a patient, we did a bowel resection, a microbiome study while I was at Yale, a guy named John Geibel. They were looking at the microbiome in the small bowel. So if you're going to do like an ileal conduit and you're cutting out like 18 inches of small bowel, well, if you cut out 18 inches and you decide to use 12, the rest of it's discarded. Okay. And the biopsy is still, that's a little bit of a different beast. So if you're going to take biopsy specimens, you have to be really thoughtful and talk to your IRB. So biosafety hazard, I think this is something that I try to do, is try to make note of communicable diseases. You don't want to, do you want to procure for patients with HIV or hepatitis? Obviously, everyone has universal consent. If you like your team, you would try to minimize the risk. So I've tried to always minimize collection from patients I knew might put them at higher risk. Maybe I probably get written up by not trying to be inclusive these days to patients who have communicable diseases, but there's not really a question that I want to ask regarding that. So unless your research question really focuses on like HIV, kidney cancer, I would, try to be cognizant of your team. Then the time points, obviously, when to obtain materials matter, based on your question of interest. I think screening, subtype, prediction, prognosis is all fine with untreated specimens. For obviously the post-treatment analyses, you need to consider time, but again, what's the optimal time? So if you're looking at like response to, a agent affecting chromatin remodeling, or like, looking at, reactivation of a pathway, really you might have to guess on the time or need to be guided by, preclinical work. So I think also trying to understand when patients are going to come back, like, are they going to come back for a post-op visit at week 1, week 2, week 3? All that kind of makes a difference. So obviously you want to know what you want to collect on. This is our list I had at Yale that we had our IRB kind of written for. And I had different time points, to annotate intake, at surgery, post-op, all this different things. Unfortunately, since I didn't have any med-oncs that really cared about anything, translational, I only really did early, probably at surgery, after surgery. So it's a kind of a limitation for what I had collected before. But again, if you have someone who's very active and motivated, like, if you're going to get an infusion, for medication, you definitely use that IV and get as much as you can get. So obviously, I think this is a strategy here, is it's really critical to understand the approach early on. It's kind of like the ER doc, who's a jack of all trades and master of none. Can you decide, can you have a little with a lot, or a lot with a little? And it's really quantity versus quality. Okay, it's different. If you're trying to find a partner, you want to kiss a lot of frogs, you'll find your prince or princess. Here, I would say that you can always add information later in the EMR, but you can't recollect specimens. So I've had always a point of trying to collect as much as possible, and we can always figure out how to maybe add annotations if necessary. Logistics. So obviously, where is the urine and the and blood going to be procured from? Is it in clinic? Is it the OR? Are tubes provided? How do they get into the lab quickly for processing? If there's a long delay, are they at room temperature of 4 degrees? You can have a fancy, what tubes you need. And then obviously, they do get lost. So have some information. or have this around, David, you might have this in your clinic still, but we had nice baggies and collection kits in different campuses on parts of clinic. So in the urology space, this would go through quickly. In the med-onc space, it would never be used and it's probably there with dust on it. Consent workflows, the patient consented, you got to figure out, they have a bag, you can give the bag to the patient, they're going to the OR, they get their pre-op labs, different types of things. If things are missed, can they be collected in the OR? How do we label things? How do we store? How do we actually, put it in the location? So I think going through each types of things, obviously urine could be stored whole, it could be spun down. A good centrifuge is great for your lab. Clearly decide if you want to supernate it or pellet. Going to figure out if you pour it off, or maybe you want to enrich different like cellular, subcellular populations. And I think things like exosome or cell-free DNA, clearly that's more important. Then blood, clearly whole blood is great for banking because it's great for genomic control. There might be some WBCs later. I've never had any issues getting it. We bank whole blood and I use that for my genomic control. You can store it at -80 and it's fine. And clearly RNA proteins metabolites can also be analyzed as well. Obviously differences in stability. Fractionation, obviously there's different tubes. We actually have a shortage of yellow top tubes here and also purple top. So, I can use different, there's no real difference between lithium heparin or EDTA. Plasma will be just the same, or the couple different SST types of tubes. And these are easy to spin down. Now, depending on the analyte question, most things, serum and plasma are interchangeable, but other times they're not. They can precipitate out and they might be very different. So obviously know what you need. I was collecting both serum and plasma, so I had, always available whatever come our way. And then obviously WBC, the Buffy coat can be spun down. It can be stored for different functional work. There are different ways to stabilize the white blood cells. There's different kits where you potentially can keep them activated or activate them themselves. Flow cytometry, obviously you need to do real time, but there are ways to have, there's a company called Smart Tube where you can stabilize and do like flow later. And then for liquid nitrogen, obviously, if you're going to bank these long term, they do better, I think, at, in liquid nitrogen rather than like -80. So it's important. So clearly, there's different kits, different spin processes. Do Ficol gradients, and you can collect the population you really you need or want. And then for procurement, you need an SOP. So if you have a procurement team or you have a pathologist, they're going to have to decide. You're going to tell them what you want, like a partial nephrectomy, radical nephrectomy. I've had times where I have a critical tumor, like I had a woman with BAP1 who had a papillary kidney cancer, and I wanted to know, what the hell is going on there. And they're trying to argue that, your SOP says you need to collect for a tumor that's 1.5 centimeters, and this is 1 centimeter. So, make sure you know what you wrote and make sure that you have buy-off, because they're going to follow as like the Constitution. But basically you have to have it signed off by the pathologist procurement team, and they'll try to follow it and every time. And then what to collect. So normal, I think the normal is very valuable, the great genomic control. Clearly, they'll have a lot of it. Take out a big kidney, you might have a lot of normal kidney. I've partnered with nephrologists to have them study like normal kidney. They've actually had we have a paper in JCI, the guy named Lloyd Cantley, whose brother you may know, Lewis Cantley. We had like 100 normal nephrectomy specimens, and he came up with a nice atlas of normal kidney. So you can have like this study of great things with the normal. We're also doing stuff here. We have a DoD grant where the normal, we're actually transforming it. My buddy Andrew Goldstein has transformed normal prostate into small cell prostate cancer. So he's doing that. We're trying to separate different parts of the nephron. It's, hopefully something that will be, useful in the future. So the normal is potentially very useful for a lot of different things. And then the tumor, obviously the tumor is not always straightforward. A chunk of tumor is not always what it seems. Heterogeneity is clearly a problem. Necrosis, fibrosis. Sometimes if you really need to have pure tumor or the best you can, I would say that you could have, you can embed it in like OCT and then, put in the cassette section, and then kind of see what you want. The last thing you may want to do is like whole genome, spend $10,000 and have it be like a crappy study. And then the fresh material obviously can be preserved in a specific way to way you want. Clearly, other things, there are many ways to process a tumor depending on the usage. Fresh processing obviously is useful for things like flow, single cell analysis, organoids, PDX. But you can also do things like, frozen analysis. Besides for genomic work, there's ways to freeze things slowly. There's ways to dissociate and freeze. This is a paper, David, this is probably up your alley, where they basically do, I think they do, CyTOF, and they're doing with dissociation and maybe cryopreserving. So obviously real-time analyses are great, but it may be difficult to do that if you don't have the staff to be collecting and processing and like doing stuff real time. And then obviously microbiome, we know this is the big thing. Urine, tumor, stool microbiome have potentially important ways to modulate the immune system. So, these are usually done by snap freezing in like isopantane or liquid nitrogen, and that's done to avoid expansion of other species. So whether you're collecting any of these materials, they get basically frozen quickly, and then there are different ways for either metagenomic sequencing or basically amplicon sequencing of that 16S ribosomal gene. And there are a lot of companies that do this for you. Our poop studies, fortunately, we don't have to do any swabs. We just poop in a bucket and send it to a company, and that's how Monty Powell does his stuff too. But hopefully, if you have to do a swab, you have a good resident or fellow to do that. Or a bad resident or fellow, it would be preferable. And then specimen plans with a coordinator. I think it's really useful to have a well-defined workflow for a project and specimens. For each specimens, you might have like, again, I have a translational kind of team. I have multiple different folks. I'm fortunate this year I got a DOD, but I don't see my future being like fully independent. So a lot of it's team science, and I'm happy to be Robin with about 5 or 6, Batmans. So like I would kind of fill this out and have an idea. Well, this tumor organoid, this tumor PDX, and try to work with our person who walks around a lot. He would just count his steps. He probably walks like 10, 20,000 steps because he's delivering stuff and working with different folks. And then obviously shipping stuff out. And then a good freezer helps you sleep at night. I think a -80 freezer with an alarm is critical. Liquid nitrogen backup is perfect. If you're looking to buy a freezer, make sure it's backed up. And then the liquid nitrogen tank, if you have access to it, things like obviously cell lines and WBC storage, Buffy coat. I think that's important for long term. This was our freezer at Yale. It was ready to be filled. And I had, it took a long time to put all these together, get everything arranged. We have Sensifoam. This is a lot of things people have in their lab for basically calling for emergency, and it goes off a lot. So I would tell you, work with your institution. You can get a camera. So if it's beeping and you see the temperature is like 79, or you're beeping and there's like a student that has the fridge open, it'd be nice. You can yell at them, shut the fridge. But in the middle of the night, if you take a look and the power's off, clearly it's potentially a problem. And then obviously the storage racks and organizations, it's really critical to make sure you have good ways to track things. And obviously tubes that are large means less samples. So when I came here, Alan Pantuk had done this trial with pomegranate. He got money from Palm Wonderful and was giving men with pomegranate juice, prostate cancer pomegranate juice. Clearly it didn't do anything other than, make them like get cavities from the sweet juice. But he had all this urine, and all the urine was collected in 50 cc's. So I have a whole freezer, like half of it's filled with like, maybe only like 200 specimens. It's kind of crappy. So I've been trying to kind of throw some of that stuff out. But, everyone argues that their specimen's invaluable and maybe they'll look at it in the future. And then clearly, you have to understand you may not want to collect too little. Like I have a freezer of 4 cc's of urine. Maybe that's a little bit too little, but I don't want to have a 50 cc tube. And then for capacity, clearly there's different sizes, but you can have a tremendous amount. You may not need to have a whole freezer. Maybe you have half a freezer, 32,000 specimens that will fill up quickly. But depends, if you're only collecting kidney, and you're only collecting your own patients, it may not fill up that quickly unless you're, like Bob Motzer and you have like hundreds of patients like a month, which I'm sure, Ritesh, you see that. But you basically organize it very well, know where everything is. So there's a clear plan of where you put your plasma, your blood, your urine, and then children. The children, basically anything you would do to the specimen, the DNA, the RNA, maybe you're looking at metabolites, so it can all link and there's a home for it. And it's useful to have them together. So when you're pulling it out, you don't have to go and look into different, you pull out the same shelf rack box. So, this was my vision, at one of the places where we had our racks, we had our boxes, we had different things collected on different areas. Organization, you could do 9 by 9, you could do 10 by 10. It's really hard to pull out 10 by 10. They're kind of get stuck, especially when they're freezing. So this one allowed a little bit easier. Just, I had mercy on people pulling things out, that it was a little easier to deal with the smaller ones. Clearly, also, you got to think of, how you're going to label things. Not every label, there's different pieces of paper. This is one over here, this is one that, is at Yale, it started breaking down. And, you never want to not know what something is. So it's expensive, this type of paper, but invest in better paper. And then obviously there's different printers. This is a Zebra printer. You could do barcoding, and some of these things you can actually use a barcode. So if you ever, don't know where the specimen is or you can't see it, the barcode might work. A lot of them will say like shelf 6, row 5, but then later on, if you take it out, want to put it back, maybe you deaccessioned it, or let's say it, falls on the floor, potentially that barcode system is potentially, I would say, useful. The organization, you can create your own. There's also freeware that's published. When I started contacting these people who built like these file maker softwares, and on the supplement said, here is a link to our program where we have freeware. And every single one, either the link was broken, or when I contacted the PI or the corresponding author, they never responded. There are institutional efforts, any CTSI institution, they have, there's this Encore software, and they use it for some of the like clinical trials monitoring. But as I learned here, we have Encore for like things, but there's a biobanking module that they have to actually pay for. So biobanking module that you have to pay for, they don't want to pay and they have their own system, then you can't. I had that I thought it was free, but clearly it's not. And then commercial ones, and our one here links to Epic, so you can pull path demographics. So if you add someone in, it can like add a lot of their, demographic stuff, I think is useful. But this is what I initially had started working with. I built like a nice FileMaker database, and there's a way to print a barcode with the specimens as well. But this proved to be, okay. But then the institution had come to me at Yale and said, hey, can you use our Encore system? We were the first one to build a bank in that system. And that's how it kind of would look. You have basically how many accruals, the 1,500 patient. We started up slow and we cranked up there. We needed to add to it. This is 2014. I would say good things about these programs. You never want to pull a specimen and then you have an empty hole. So deaccessioning, I think, is useful if you're ever going to buy software. I think it's important to have something to say next up, because you don't have a separate database and say, hey, which one am I on? And then you forget. So the database will know what's next up, and the database will print the label. The child specimens, I think, are useful because you want to know where each sample's DNA and RNA is. The online backup, I think, is critical, and one that's online that allows you maybe to have a student who might be virtual, or someone who has COVID who can't come into the office, to kind of add specimens. And the printing label, again, is useful. But always worry about upkeep. Like right now, I have to pay $3,000 a year for our system here. And I guess the invasion of Ukraine somehow increased the price this year, which I don't know how, but I can blame Putin for a $500 increase. And now our biobank, obviously, if it grows, it might be successful. We have this as a resource, and we've started to we had no kidney cancer grants before, and past 3 years we've had a bunch. We've had a DoD idea development, 2 concept awards, a KCA award, a lot of this finding good collaborators. And I think the control gives you some flexibility. So, you don't have to, like Abhishek, you don't have to go to the urologist and say, hey, can I use your freezer, or your pathologist? You can say, hey, I've worked and I have these specimens available and I built my tissue microarray. And they're like, how'd you do that? Well, I planned and I did it through all the mechanisms. And again, we've had a bunch of people that weren't working on kidney cancer because they really had no, champion, they had no access, and I think it can grow. I think you will have to build like a constitution when things get busy, but a lot of the places, if you're the only one or there's only a couple, you have years to think about that. So that's my 2 cents, and happy to answer any question, guys.
That was great, Brian.
Right. Sorry.
One thing that I've noticed now with like grants and things like that is there's a lot of inherent costs that I never think about. I mean, just to like think about like some of the stuff that you've put together. Do you want to maybe talk through a little bit of like what are like the primary things that you would sort of build into a budget?
Yeah. I mean, again, a lot of it's staff costs, because I'm lucky here, like for philanthropy, that pays for someone's salary, but you have to that person's salary, you have to worry about like liquid nitrogen, you have to worry about like access for like plating wear, access to pathology costs. Like they charge me $30 every specimen. Initially, what they're doing at Yale, I was doing a project on heterogeneity, and we have a paper in clinical cancer research where we did sequencing of different regions, and they were charging me $30, $30. I'm like, it's the same freaking specimen. So, those costs kind of add up. I think, and obviously, for your freezer or upkeep, or, again, the freezer is $25,000, but, you might have a service contract on that too, which is a couple $1,000s. And
right.
The biospecimen might be $3,000. You might have to budget a student to enter things, say, hey, I'm going to have 10% of I'll have a student's time, and they're part-time, and I'm paying them like, $20 an hour. All this stuff kind of adds up. Ask for more. And then, yeah.
I think the big challenge, I think that we have faced is really not so much the access, right? The access is largely determined by the clinical volumes also. And sometimes if you have a good partner on the clinical side and the volumes are good, you can get material that you need. But I think the bigger challenge tends to become what are the questions that you want to answer with that material, right? You can't just indefinitely keep storing them, hoping that one day there will be a question for which you will utilize the sample. So, and when you have a question, you can't go back and start collecting samples at that time, because then it takes like 2 years for you to get where you want to be. So I think, and we discussed this on this in this group before, was to have some sort of a centralized information / material sharing sort of a agreement, so that everybody could be on the same page about what was available, how they were doing things. I don't think, I think in fact, Brian went and tried to see if legal teams at different universities could work something out to that effect, but turned out that that was not a possibility. I really still feel that if we have a way in real time to start sharing, especially technical knowledge, right? So how do we turn the renal mass into an organoid or into a PDX? I think that would be invaluable knowledge. And no matter what we see in papers, when we actually try to do these things ourselves, the success is never quite what is reported, right? So these are the types of things I think would be like not hard material, but rather knowledge sharing itself would have massive appeal, I think, for the community to progress and make whatever discoveries they need to make to push this forward.
Yeah. I mean, I wish we have a Southern California consortium here where Monty Paul started a 501c, and we've been trying to look into it, like a master agreement with that master umbrella, an organization called like the GU Central, whatever. And we're going to even sharing like clinical stuff with that one organization, and then everyone would be working. Just the legal stuff, it was just such a headache, we kind of gave up. But that's obviously be so much better to, learn from everyone's mistakes. Like making an organoid PDX and that technical stuff is obviously that could be easily shared.
Exactly, and then also think about the other aspect, right? So even at institutions where these resources are available, and we're trying to do this at the Cleveland Clinic now, but I don't know how far it will go. But let's say you cut out different sections of the same tissue and give it to different investigators, and then they all go and do this. Let's say all of them go and do RNA-seq. What's the point? You just spend 6 times the amount of money by sending it to 6 different people, when instead having a central repository and a database could have been so much more powerful to advance the same.
That's where I do think, if the person who's coordinating, like I know who's going to do what with the different samples. But like I know we have an HLRCC organoid, and I know that patient has an HLRCC PDX for one of my patients. So I do think it's one thing if someone's just giving out samples and not knowing, but if you have someone who kind of has an idea who's done what with what, it becomes a little bit tricky. But that's why I think you have to have like research meetings and know what other people are working on.
Yeah.
And we have a list of like what went where. So if you could, I think that'd be, a great way to kind of not reduplicate efforts, but even in the same institution, it's hard.
Yeah, we've done something here. I mean, one thing that also we found, which has been helpful, was we use like, we've kind of spearheaded like a REDCap database so that patients are almost like de-identified or even given like a specific unique identifier already. And so we sort of try to log similar to how you do it, Brian, with like specific clinical data. And then also like, is there a PDX that was generated from this or organoid or cfDNA or whatever, so that somebody that can then survey a database and say, if they have a specific question there, they can see if there was like a model developed from a specific patient that they're looking at.
I mean, that's great. I mean, it's great you have enough people doing different things. So, it becomes great to have a centralized resource of what's done what with what specimen.
How have you all dealt with the authorship issue? So one thing with like creating a REDCap database that we have faced is the clinicians who are depositing it and actually updating the charts, they want to be then involved in like the paper and like writing the story and all of that, regardless of what it is actually being used for. And so this created like a whole thing about like, who quote unquote owns the database, and I just don't know how to navigate that part either, because I don't know who's putting the information in there, and I don't know how to credit that person for that information at the back end. It would be interesting to see what you guys are doing to fix that. Have you all faced these challenges?
I guess at least here we've tried to be, as collaborative as we can be, with the data that we're sharing. So we haven't, I don't know if I've faced that so much sort of head on. I do agree though, like there's a lot of stuff that we'll send tissue to one place and then I don't really see the data, until it's been like sort of in the refined format, I guess, at the very end. But yeah, so curious to hear about others.
But Paul Russo's not doing the RNA-seq on the tumor he removes?
Well, I was going to ask you, Brian. I mean, the one thing that I think that, obviously Ari does a lot of the spearheading for us translationally, but I think one thing is always that it's, for some of us that are on the med-onc side or even on the lab side, it's always interesting to try to hear about patients who might be going to an operating room and would be like critical samples to kind of get, and it's hard to kind of make sure that we have the right workflow in place and stuff like that personnel wise. I feel like a couple of years ago, even it was very, very complicated to even do that. I think we're getting better at it, but we're still not close.
I mean,
yeah.
well, I think you need to have people invested. The problem is, if you have 8 surgeons and one person doesn't give a shit, it's a problem. Or, if you have Ari who gets the specimen out and says, please send that to the lab right away. And then you have, Paul Russo who is sitting back and they put it in the freezer overnight. Meanwhile, the RNA is like dying. And then Martin and you get a specimen and it's shit because, it's all degraded. It's really difficult. Or you're really hoping a patient got consented and then, they scared the patient off. Or you have a partner who has their own protocol and they banks it themselves. So you have to have everyone invested in, and I've had really people who've been very motivated and other people who are not. I do want a lot of patients who are starting systemic therapy, and David, Harriet and Mario are wonderful, but they were not going to go out on a leg to do much for others. So I know if I had a guy like David, we would have been more successful, but he came a little too late.
Come on back.
Yeah.
I need someone the urology side.
Yeah. So again, it's hard to find. Again, I think Abhishek, if you find the people who are motivated to help fill that database in, and they can go back and continue updated, I think consider them a good partner. And I don't think the people no one wants to see themselves as a database filler or a procurer of tissue. So I think if you can empower people to feel like they're really participating, whether it's authorship or or have the potential to add additional value, they'll be more invested.
Yeah. So what I have done now is I have told them that how about we have like a common clinical research coordinator who I will pay for through my grants, and then that person's job full time should be just consenting and managing the database and dealing with all, like basically become the bridge between the clinicians and me, and because they don't have to pay for it, they're okay with that idea.
Yeah.
And so especially I think the lapse that we faced, especially after Brian in fact left from here, was just like we have this like 5-year gap in annotation and the entire thing. And so continuity is also a big challenge when you have people who are not directly invested in the entire process, so yeah, I agree. I think the more we can keep them engaged, the better this will be in the long run.
Yeah.
Do you find any ways to sort of improve, at least I guess now thinking about like the contracting end, ways to sort of make things as broad as possible so we don't have to sort of renegotiate contracts, and like even I find that it's like almost easier to just negotiate like the de-identified data piece, right? Like if you generated, Brian, a lot of RNA-seq data or whatever, we can share that, then like it's almost easier than just sending the tissue here, right, for sequencing or whatever. And I feel like we still run into like renegotiating over and over again the contracting for that, and that's what delays.
I would just be as broad as possible on anything. Any specifics kill you, because then someone will comment and you have to go back. Every time you send something illegal, it's like months and months and months. We have a trial that I'm trying to, we have a trial based on the lab research that started with at Yale and here for HLRCC. And, it's a PARP inhibitor TMZ trial. And it's been a year trying to get the NCI on board. And it's just been horrible. And every time I have a lawyer touch it, it's like 2 months before they come back with changes. They're not invested. So, again, every little minutia they'll find and they'll say, what is this statement here? I want this changed. So try to do, especially any banking, just make it broad. Not going to say like, we're going to profit on selling your body parts and tissue. I mean, you got, if you make it that clear, no one is going to say, hey, I didn't realize, that my tissue is going to be like sold.
I feel like that's like when we first started doing all of these talks and everything, like I think that's what Abhishek was sort of saying, right? We sort of talked to Brian to say, it would be nice to sort of grow this like consortium as you're almost doing right in California to like, RNA-seq data that people can then go into, or like our own version of like a TCGA, right?
Yeah.
Where
So,
people
I
can
mean,
sort of-
Monty's tried to do that by having a 501c and trying to have that organization have agreements with each UC. So like
Right.
Rana's trying to work with it, Sia Danishman, Young Kim, and we're trying to work on that. But it's going to be it still is going to be a headache. But again, it's one organization with different, so if I have to, I don't have to do a contract, meet a USC, meet a UCSD, meet a Cedars, meet a City of Hope. It's just like one master uniform agreement and
That's
everything
cool.
shared with that entity
Huh.
for specimens or clinical data. But easier said than done.
Yeah, seems like it.
Yeah. So,
Yeah, I think there's a lot of places that unfortunately, something getting done and then not getting the IP is much worse than, it not getting done at all.
Yeah.
So I think
Yeah.
yeah.
Goes,
yeah,
right?
I mean that I had a whole contract with the Rebiotics company with delivering poop to them, and the lawyer was like, they were so focused on discovery. I'm like, I'm literally giving shit to the company. There's going to be there's not going to be any exciting IP that comes out of the shit for my patients other than it's shit. So yeah. Yeah, and I'm like, I don't know how much IP is going to be like, okay, Achomenertia. Okay, that's a good species. I'm not going to like discover some, yeah. So,
Yeah.
no, but I got to make a poop emoji, which is good. Yeah. So got any questions you guys have, shoot me an email, and I've made a lot of mistakes, and, I think, I'm still learning myself.
Thanks so much, Brian. This was really helpful. I might actually bug you a little bit later, just as I'm trying to get a fresh tissue bank for kidney up and running.
Well, why don't you use the one we have here? I mean, again, yeah.
I'm going to use, I think, that same protocol, but kind of working through where we can actually collect it, so it's not the 4 hours until it gets to search path room and all these sorts of things. I'd love to.
Yeah.
Deal specific, but I'd love to